Now, I just want to say a few brief words about the process of fermentation. And the reason I talk about this is, you may remember when I talked about the respiratory control, I noted that the absence of NAD and FAD would stop the citric acid cycle. Well, similarly, the glycolysis can be inhibited by the loss of NAD, but glycolysis is a very important system for the cell to be running. And so cells tend to avoid complete blocks of glycolysis unless they have a lot of energy, okay? So what I want to talk about here is the way that cells keep glycolysis going even when they don't have abundant NAD. So what this process is that I'm going to describe to you is a way for cells to make NAD for glycolysis. Well, why do they make NAD for glycolysis, or how do they make NAD for glycolysis that are different from what making NAD for the citric acid cycle is about? Well, remember that the citric acid cycle is occurring in the mitochondrial matrix, and that glycolysis is occurring out in the cytoplasm. So they're physically separated from each other, and NAD and FAD cannot cross the mitochondrial inner membrane. So NAD that's made in the cytoplasm is going to stay in the cytoplasm, and it's going to help glycolysis. Okay, well, let's consider the breakdown of glucose to make products. We've talked about this before in the metabolic pathways. Glucose, the sugar, which contains 6 carbons, is broken down in glycolysis to make 2 molecules of pyruvate. And at that point, those 2 molecules of pyruvate can go into 3 different directions. And the directions that they go really depends upon how much oxygen is available or how much the NAD is available, okay? And so, well, in the normal situation, when we have respiratory control and so forth, NAD is related to whether or not oxidative phosphorylation and electron transport are running, okay? So, well, when will cells run out of oxygen? Well, cells that are, if we have cells like muscle cells that are exercising very heavily, for example, one of the things that can happen is that they can use oxygen faster than it can be delivered. Well, if they're using a lot of oxygen, they need a lot of energy. They don't want to shut everything down. If the citric acid cycle shuts down, then the cells become totally dependent upon glycolysis for the ATPs that it can generate. And so this process I'm going to describe to you then is how that cell basically stays alive in the process of its metabolism. Well, glucose goes to 2 pyruvates. There are 3 different directions it can go. When oxygen is abundant, we see in all aerobic cells that the pyruvate is converted into acetyl-CoAs, and those acetyl-CoAs go into the citric acid cycle. They make abundant NADH and FADH2, which is, of course, converted into ATP, and everybody's happy. When oxygen becomes limiting, however, there are 2 different paths that pyruvate can take, and those 2 different paths that they take depend upon what kind of cell that we're talking about. So the first cells that you're probably familiar with, when they run out of oxygen, they go through and they do fermentation. And by the way, fermentation is a process that occurs where pyruvate is converted into other things in the absence of oxygen. Now, I say that because people think of fermentation, they think, oh, making of beer and so forth, but that's only one kind of fermentation. I'm going to talk about 2 different kinds of fermentation here. The first fermentation, of course, is what's used in the making of, by brewers for making beer and wine. In this case, we have yeast cells. And yeast cells, when they run out of oxygen, they need to make NAD to keep glycolysis going, okay? Well, how do they do that? If in the normal scheme, of course, they go through the electron transport system and produce NAD, but if electron transport is slowed down because there's not abundant oxygen, then the cells have to find another way to keep that going, because notice up here in glycolysis, 2 NADs are needed for every cycle of glycolysis. So it's important then that fermentation provide NAD, and we can see that actually happening here. Well, in fermentation, what the cell is doing is it's converting pyruvate, this is being a yeast cell, is converting pyruvate into ethanol. And the byproduct of that is not only ethanol, but carbon dioxide. So when you are brewing something, what you're doing is mixing up all these yeasts, letting them metabolize things, and you're capping it so that you prevent them from having oxygen. When this happens, their only way to stay alive is to keep doing this cycle here where they are making NAD, and a byproduct of making that NAD is making ethanol. Well, that happens in yeast. What happens in human beings? Human beings have situations where, as I said, they might be running faster than their body can deliver oxygen to muscle cells that need it. And rather than have the muscle cells die every time you exercise heavily, muscle cells have a different kind of fermentation that they perform. The muscle cells in animals go through a process where the pyruvate up here goes and instead of being converted into ethanol, is converted into a molecule called lactate or lactic acid. The byproduct of that is just like it was for the production of ethanol, NAD, and that NAD is then used to keep glycolysis going. So, animal cells go through fermentation very much like yeast do. The products, however, are different, lactic acid instead of ethanol. Now this is shown schematically here a little bit more clearly, in which we can see the reaction of glycolysis that needs this NAD. You may remember, first of all, that in glycolysis there's one oxidation. The oxidation reaction is the conversion of glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. That reaction absolutely requires NAD because it is an oxidation. So we can see here on the left the NAD going in. We see the reaction occurring and the production of NADH. If this NADH is not recycled, the cell's in trouble. And so when electron transport is not available, as I noted, fermentation takes over. And we can see in this case that the NADH is converted back to NAD by the process I just described involving the conversion of pyruvate down here over to lactate over here. As a consequence, NAD is regenerated and glycolysis can keep going as a result of this cycle between glycolysis and fermentation. A similar process occurs in yeast, and that process that occurs in yeast is shown here. Again, we see glyceraldehyde 3-phosphate being converted to 1,3-bisphosphoglycerate that converts NAD over to NADH. The NADH is recycled back to NAD. And in this process, we see a couple of steps happening. We didn't see both of them depicted earlier. Here we see that pyruvate, which is the end product of glycolysis right here, is converted first to a molecule called acetaldehyde. And this reaction involves loss of the carbon dioxide. This is where the carbon dioxide is produced. And that acetaldehyde is reduced to ethanol. Well, that reduction, of course, takes electrons from NADH and makes NAD. So although the process is a two-step process, and the process that occurs in us is a one-step process, nonetheless the outcome is exactly the same. The cells produce NAD as a consequence of the fermentation process that's going on. That NAD is used to keep glycolysis going, which generates NADH, which keeps fermentation going. So again, we see this cyclic process that's occurring when the cells are out of oxygen.